So welcome everybody to our September AnVIL Demos. Today is September 17th. Today we're going to have a presentation from Paulo Lyra from Moffitt Cancer Center. He's going to be sharing a demo in the first half of our meeting today about Galaxy learning and modeling, machine learning, accessible and best practices. So we're really excited to hear about this today. After the presentation, we'll turn it over into a Q&A. So we can ask Paulo questions about his presentation or any other questions that you have about AnVIL. So without any further ado, I'll hand it over to Paulo and you can take it away.
Okay, thank you. Thank you for having me. This is a really exciting moment for me to present my work. Hopefully I can show some interesting tools to work in AnVIL and Galaxy. Okay, let's get started. So today I'm presenting this combination of AnVIL, Galaxy, with some tools that we are generating in our lab from Dr. Gak's lab. We are working with some awesome machine learning tools for Galaxy. And for this presentation, I'm going to focus on a GTEx data sets that we started using to confirm that our tools are doing a great job and doing what we expect. My goal today is to show how to apply machine learning models to the public GTEx RNA-seq dataset to advance research. And I hope that this gives you some interesting ideas to accomplish other tasks or goals that you might have. Let me start off describing where these tools that we are developing came from. So a lot of people now are using artificial intelligence to do research. So if you look online, you're going to see a bunch of publications now involving machine learning models to predict patients' response or treatments, etc. And a couple of changes. If you're not familiar with machine learning models, you're going to face some barriers to start doing that research. And I'm listing here some of those challenges, which is first, you need to understand what softwares and libraries are available to you to use. And that creates the first barrier, because if you're not familiar with command lines, libraries, and import things, running code, this will be complicated for you to start doing those researches. Another thing that we also see that is really common is the computer resources, because people usually work on their computers that are not powerful enough and the data sets are big. And this is impose another constraint for developing work with machine learning that requires computational resources in a great scale. And another thing that it's really important that we notice when we are looking at the papers that are published, they are great, they show results, but they are not consistent between publications. So usually you're gonna ask questions about the model, how they perform, and usually the authors of the paper are focusing on something specific, and you have another questions about the model, and this is not easily to get. And this is what we are calling best practice, how much transparency on building those machine learnings and making them available for the readers after you publish your paper. So this is another thing. And finally, it's what we call full reproducibility is, again, this transparency, if you have access to the whole setup parameters, the model files at the end, if you have that at hand to build again that model that was published. And it's really interesting to have that in many cases when you're seeking other questions or want to develop it from that specific model and further. So okay, so that's our first glance of challenges that we are trying to overcome. And we came up with this project, which is called Galaxy Learning and Modeling, which shorts for abbreviation for GLEAM. And this GLEAM is the next generation of Galaxy framework, providing specialized tools for machine learning. So GLEAM, it's the concept. So GLEAM has some core values that we build on top of that. So we are looking for integration, having everything together packed in one place, end-to-end integration to simplify these tools. Accessibility, so anyone, it doesn't matter if you're starting learning machine learning or if you're like advanced user of machine learning, anyone can come and use these tools easily, and they use their strength for their research. Best practice, because we are trying to be as much as possible transparent, showing everything that we can and making everything consistent. So same results, same display for anyone that uses this tool. Reproducibility, because we are exporting all these files that are created after the run, including the model files, the report, everything that the artifact that is generated for that specific experiment is going to be available, and then you can share, and this brings the reproducibility at hand. And finally, the scalability, which is really important. And I mentioned that sometimes you have big files, big experiment, and this is really important to be connect with some powerful resources, and that's where AnVIL comes along, because it provides this cloud environment that you can choose the sizes for your resources, which is really great. So this is GLEAM for everybody. Just a quick glance what's happening, how we are building this. So as I say, GLEAM is the concept, and we have three different tools that are embedded inside this concept. So one is called a tabular learner, image learner, and multimodal learner. And the idea here is to provide the user a clear input when they're going to use each tool. So the names are self-explanatory. So we are leveraging different libraries underneath the hood. So we have PyCarry for the tabular learner. We have some good experience with that because of rapid experimentation, automated pre-processing, etc. We have Ludwig for image learner for deep learning models, which is really great and powerful. We have some great pre-models that you can use, select from. I'm going to show you in a bit. And multimodal learner, which is probably what's coming next, because people usually don't have a single modality, like a one format of input. Sometimes you have images and tabular data and text. Those are the three targets that we have for this tool. So in case you have like patient's history that it's written by a physician, you have the images from this patient, and also you have the clinical data that it's tabular for this patient, and you want to have that in a model to predict something, some target that you have, then you can come and use this multimodal learner for your research. So these are the tools that I'm going to show you today, like the first, the tabular and the image. Multimodal learner, it's under development. It's like 85% complete now. So soon we're going to have examples of that too. Happy to come back and show that. So as I say, we have best practice embedded. So just to give you a quick look at what it's best practice embedded here. We have a bunch of data, plots, and tables that makes easier to understand the machine learning that you just built. And you can also access your model to be sure that it's doing its work, what it needs to be changed, or if you can, you're ready to deploy that and use in the real world. So we have four tables, 9 figures for image learner, and 4 tables and 15 figures for the tabular. So this is a great a number of figures and plots and to make them transparent. Okay, so we have those tools available as now in 3 different places. So AnVIL Terra that I'm going to show you today, also Use a Galaxy and Cancer Use a Galaxy. Those 3 places you can find the tool. If you look for using the search tools, you can type learners and you're going to see them pop up right here. Okay, so the first project that I'm going to show is our probably the one that we test to make sure the tools are okay doing what we expect. So we are going to build a model that predicts the tissue origin based on the RNA-seq. So we take this following approach. We take the GTEx data sets from AnVIL, import that to Terra, and we perform 2 tasks here. The first one, we perform a transformation from tabular to image. And this is for 2 main reasons. The first one is because we want to test our image learner tool. So we need images to input in that tool. And the second one is in the past, we learned that Google used to do this transformation from tabular data sets to image because they had tools that only took images for their models. So it was a great experience to do this transformation, and it's also good if you want to have some visualization of your data set. Looking image with some dots on it, it's better and gives you a full overview of the data set instead of like a big table that it's complicated to see end to end. So that was a great, really interesting experiment. And then finally, we used the tool to generate the convolutional neural network, the deep learning model to predict the tissue origin based on the RNA-seq. So we have 3 main steps here. As I mentioned, select the GTEx, build the images from the tabular data set, and then running the tool. If you're not familiar with AnVIL, that's the outline that I drew to make it easier to me to understand. I hope it's also helpful for you. So the components are kind of linked together, so you just need to understand how they link together. So AnVIL Data Explorer, it's where you search for your data sets available there. In this case, you're gonna see me. I'm gonna show you live what I did. So I went to AnVIL Data Explorer, look for the GText available data set. Then I ask to AnVIL to send this to my Terra workspace. So now I'm linked to this data set. So I can work on my own workspace and do what I need to do. Then I'm gonna launch a Jupyter notebook where I'm gonna pre-process the data, do the transformations. And finally, I'm gonna launch the Galaxy project, the Galaxy environment, so I can use the learner tools inside Terra. So that's kind of the whole picture from this experiment that I did. So let's see if I can show you how I did this. So I went to AnVIL project, I scroll down and look for data sets right here. I'm gonna open the AnVIL Data Explorer. And since I'm looking for GTEx, I just type GTEx for you. And you have this public data where you're granted access by default. So that's why I went for this one right here. Then I just click export, analyzing Terra because I want to connect that to Terra. Well, here you have the option to select all the data you want to import. I import everything at first because I didn't know exactly at the beginning what I need. So I just export everything. and I request the link. And this is going to give me the link, and I'm going to open Terra. It's going to show me the next page. This is where I'm already logged in, so I have my log already here. So it's going to ask me two things. Do you have a workspace already there that you want to link to that workspace, the data set, or you want to create a new workspace? So if you want to create, you're going to fill up some information here and then create that workspace, or you can select the existing one if you have. So I have one already. So this is going to take a little bit of time to import, to connect everything. So I have already here the page with the linked data set to my workspace. So let me show you. This is my workspace, just to confirm for you that I'm seeing my workspace right here. And here is the AnVIL Connect to my workspace. Okay, so now I have this to work with. Here is the dashboard for this data set that I have. I'm going to come back to this storage details right here because as you can see, there is a bucket name. This is where it's like a storage that I'm going to use for everything that I'm going to work and generate, because I'm going to generate some data sets. So this is exactly what I need at some point. I'm going to go back to this later. Okay, I think for this part, it's okay. We can skip. I have this all in my slides too. I don't know if you make these slides available, but if you want to see, like, remember that, I can share my slides. It's okay. So just to give you the perspective why I'm doing these transformations and what I need to run my tool. For the image learner, I need two data sets, two inputs. The first one is the metadata input. This one is going to be a tabular, a CSV file that does two things. The first one, it tells the image path. So it links one image that I generate, so it has the image name there. This is one column. The second column that I need to have is the label, because that's what we're going to use to train the model. This is the ground truth that we're going to use. So you need to have those two things on your metadata input CSV file: the image name, the file name, and a label. That's all you need. If you want to have a third column, which is the split, meaning how you want to divide your data sets into training, validation, and test, you also can have that. Those are numbers. 0, you're saying, okay, take this sample and put in the training. If you put 1 in that column, you're saying use this as a validation. And if you mark as 2, then you're setting to the test set. So, okay, I can have that. This is optional. You don't need to do that, because our tools, if you don't have that column, we're gonna do that for you. It's just in case you want to select that for any reason. And then the image input we are requiring a zip file. So you're gonna pack everything, all the images in one zip file, and then use that as an input. Okay, another thing that it's important here that I'm showing is to know where to find the file. So if you go to the page right here, data, inventory, you're gonna see there is a file reference right here. Remember that I told you have a bucket, a storage? This is the name that the computer knows which file you need to select. So in this case, I'm looking this to know which file I want to use. And as you can see, there are a bunch of files, and I'm looking for two specific files here. Let me open again. So two files that I'm looking for. One, it's called this long name, gene TPM GCT GZ. So here is in rows we have the genes. So all the genes that were measured, the RNA-seq that was measured during the experiment, the machine part. The columns are the samples. So you have this matrix that you can work with. And it's a long, as you can see, it's a long file with a bunch of them. And the cell that you select, this is the number of transcripts per million for that specific gene in that specific sample. So we have that in place. I'm looking for that in this list. I got the file reference. That's another important thing to remember. So I got this. And I look for a second file. This file is the sample attributes where the metadata is storage. So I can look for rows. Those are the samples. So now you see the match. I have samples in the first file. Can I match in the rows of the second file? And the columns here are the metadata. And I'm looking for a description of that sample. And because I'm working with origin, I'm going to look for the column that says where this sample is coming from, what tissue. So this is builds my my ground truth in the metadata input that I need. Okay, so I'm not going to go over the whole script that I wrote, but just to let you know the steps. First step, select the tissue that I want to work. There are a bunch of tissues at this moment. I just select a couple of them. Again, it's just a proof of concept. So I have brain, heart, muscle, etc. Some of them. I took only 200 samples from that. I don't want to overdo this. It's just again a concept that I'm trying to prove here. And I did exactly what I described. I iterate over one file, find the sample names, and then I go to the second file, iterate with those sample names that I found, the 200 that I found. I iterate, extract the sample's tissue origin, and that's it. I'm done with that table. And finally, I took the whole gene expression that's in the column, if you remember, and transformed that into pixels. So each pixel represents one gene expression. And it's a grayscale, so dark means low expression and goes up to white, and this is a high expression. As you can see, you have like a really nice representation of the table. That's why I'm saying it was a really interesting exercise, because you have a visual representation of the gene expression. Okay, so to run the code, I have right here the code already. If you want to have access it also, it's fine. What I did is I start one environment. Did I stop sharing? No, it's still sharing, right? Okay. So I started a Jupyter environment. I went to settings. Here, it's really tricky because you need to test some parameters to see how much resource you need. So make sure that you're testing. Sometimes you're going to fail because you put in less resources. You don't want to go to over resources, because that makes you pay more, but you want to find this sweet spot. It's just testing, yeah. So for this one, I select the Python environment already here. For me, work with 8 CPUs and a memory of 52 was fine for me. Less than that, I had some issues because the data set was not completely loaded. And then I hit the button to start the process. Because it takes a little bit of time, I went ahead and just started beforehand. So once this is green, that's good to go. I went here to the terminal, and I can see the terminal right here. What I did, it was just like, I use Vim, people use Nano, then it's up to you, but I use Vim, and then I create a file, preprocessing.py, and I just paste here the code. And that's it. What I need you to do, if you're going to do this after, is just to come to what is it? Confirm these files to make sure you're selecting the right files. Remember, I point to you that column that shows the file reference. And also, I need you to come here in the bucket and put your bucket. Remember, if you go here, you have the bucket address. So you just copy from here to here, and that's it. Right here. And that's it. Everything's connected. You're gonna run this code. So I'm gonna save. Here is a column WQ to save and close. And I just Python pre-processing, and this is going to start processing the whole data set. What's going to happen is this code is going to output everything to my bucket. So I don't need to worry about that. So again, this is taking a long time. It's a long file to load, and it's going to do a lot of processing. I'm gonna skip this because of the sake of time. Once you have this completed, then you have the data set ready for Galaxy. Same thing, I started the environment, we went here, setting the environment. Once again, you need to test to see what kind of resources you need. Try to be as much precise as you can. For me, work with the CPUs of 16 and memory of 104, because again, this is heavy model training, so probably you need some more room at this point. So I start the environment ahead because again, it takes a little bit of time, like a couple minutes. I have here the Galaxy running, so I'm gonna open Galaxy. And this is where it gets really interesting, because you don't need to do anything to connect Galaxy to AnVIL, because the buckets are already connect to your environment. So I'm going to select the image learner right here. I'm going to import the data. Remember, this is on my bucket, right? So if I choose remote files, I have AnVIL right here. Remember, this is the name of my workspace. I click here, I'm gonna see folders, and I save on other data files. And here are my outputs. If you do the same thing from that code, you're gonna see exactly the same thing. So this is the pre-processed data sets. So I just click here to load the files, start it, and that's it. It's going to load. That should be quick. I hope so. And here, as you can see, I go ahead and it start to explain some of the tools. So this is the image learner. Again, you're going to build deep learner models. And as you can see, it's as simple as possible. So you have the metadata containing the information. You have the zip file. You already read that you have a zip and a CSV, so it places for you. And now we can select different models. We have a list of models based on the PyTorch library. So you can have different deep learning models at hand. It's just click whatever model you want to run. So here I'm gonna run a ResNet 50. You can use pre-training weights or start from scratch, whatever works for you. We have augmentation, which is if you were working with, for example, digital pathology, you have those stains, those marks, and you want to have more, avoid overfitting. So you can create this augmentation to flip the image, change the images so the model learns different aspects that the image can come from your data set. Okay, so that's it. That's all you need. Just click there. Let me see how many clicks I did. 1, 2, 3 clicks, and that's it. I'm gonna run the tool. And now, as I mentioned before, we are trying to be transparent. So you have 3 different files. One, it's the report. The second one, it's the model file that you can share, and some artifacts that the library can generate. So you have everything Actually, those files are exactly what we are consuming to build the report. So those are the raw files, I would say, if you want to look the raw file instead of the report. So again, this is a model training and testing that takes a couple minutes too. So I went ahead and I have this done. So this is what you're gonna see. The files all green, everything you can download, just clicking download here. And I want to show that our results are really good for this experiment. The model is performing really well in the training, validation, and test, like almost one, which is really good for all these metrics for accuracy. Loss is like the only number that it's when it's low, it's best. But the rest is like one, it's the greatest, meaning we expect this. So this is not a surprise because the tissues express different genes. So we know there is a difference of signal in those tissues. So this confirms that our models are able to do the distinction and predict which kind of tissue the image is coming from. So again, showing the plots. The plots are to use to assess the model, see if you can improve anything. You have a bunch of metrics from the test. So I didn't mention, but this is trying to be separate so you know what you're looking for. Test results, training validation results, and config and results summary, just if you want to skim through the model as a whole. So this is plots. You have confusion matrix, classification report to see how they behave separately, and so on and so forth. I don't spend too much time on that, but that's how you use this tool on AnVIL. It's super simple like that. We also have the tabular. Let me see, where is it? My Galaxy. Okay, I didn't open. I have my Galaxy right here for the second tool, which is the tabular. This is running. I already have the tabular. Okay, let me just give you where this is coming from. Let me skip all the steps again. If you have the curiosity or want to remember what I click, it's on the slides. So we have a second tool, the tabular learner, which is the classification now using tabular. So we just need one input. And for this processing, it's just taking the selection of tissues we want to predict. And here we are predicting age. Sorry, forgot to mention that. So we are trying to predict a different thing, not tissue or origin anymore, but age. And this is kind of more complex. So we are now navigating this area to can we predict the age of the patient based on gene expression? So it's a really interesting question to look into. So, okay, so I iterate over those same two files, extracting the RNA-seq, went to the metadata, extract the age of that sample, and that's it. I have a table like this at the end. So I just plug the age in the gene expression of the sample. Okay, so that's what all I need for the tabular. And our results are kind of interesting because since we are doing prediction, we need to pack the age into groups. So 40 to 50, 50 to 60. So those are the packs of groups. And if you look at the results, the confusion matrix here, you can see some packing, some problems of the model to identify those ages as a group. So we are looking to if we can probably shift the way we are grouping the ages instead of a tens. Maybe biologically, by biology, we can group people not by tens, but something that is more biology related. So we are looking for references, publications that might gives us this how group people by age, not by tens. But this is a really good experimentation to start our project. And this is the work done by Isis in our lab. And that's how we are developing this AnVIL project. So just to finalize, we are testing the feasibility of image and tabular learner in genomic data. We demonstrate that we can do this in Terra Galaxy in the manner that it's easy for us to work with. And probably this sets us to a framework that can be extended to other projects based on all these resources available in AnVIL. Thank you so much, and happy to answer questions.